chromosome immunoprecipitation, or chromatin immunoprecipitation. And I put this in the bonus materials. I mean, it's like 20 pages long. Obviously, I'm not going to ask you any little details. Most of this you don't need to read. But especially the introductory thing, or regions, and they kind of describe the general thing about quantitative or chromatin immunoprecipitation. That's fair game, okay? So at least read the first 4 or 5 pages of that. Okay, here's some review materials from 431, although I could put a picture from your textbook in it just so I can at least say to use your textbook a little bit. Okay, so DNA in eukaryotic cells is in the... That's right. Or nucleus if you want to speak with southern accent. Okay. And in order to get all of that DNA in the nucleus, because you had to package it, right? And this is done by wrapping the DNA around these proteins that are called histone proteins. So that picture is not very good. This picture is actually an improvement, where there's basically these 4 proteins. There's 2 H2s and H3 and H4, and then you get 2 of each. So there's like a pair of dimer of them. Or dimer of tetramers, actually. And the DNA wraps around it to form this beads on a string type of structure. And this is just done so that the DNA can be packaged efficiently, because once you get these nucleosomes, then the whole thing will pack up like this in a tight chromatin. Okay, so I left this from 431 lecture. DNA is acidic, so what amino acids do you think are abundant in the histone proteins that made them stick to DNA so well? Here's a hint. They make a whole special point about pointing out about the abundance of lysine and arginine. Okay. So I want you to just think about this. This chromatin, this tightly packed stuff, is in the nucleus. By the way, if you have twins, so that identical twins, usually if you know them well, you can tell that they're a little bit different, right? There's some small differences between identical twins. Okay. Any differences that are between them are likely due to how that DNA was packaged, how small events took place early in their development, right after the cells split from each other, that caused the DNA to package just ever so slightly, so they had a little itty-bitty difference in gene expressions. Okay, so what's that tell us? What's important about chromatin in the packaging of DNA? It's going to influence which genes get turned on, right? In order to turn on a gene, we have to make the DNA accessible. So what do we have to do if we want to make the DNA accessible? We need to sort of unwrap. That might be too strong of a term, but we need to maybe move some of these nucleosomes or some of the histone proteins out of the way a little bit in order to make a stretch of the DNA more accessible to RNA polymerase and the transcription factors that will recruit RNA polymerase to the particular gene. Okay, so if the histone proteins are attracted to DNA because they're positively charged, what can we do to those proteins in order to make it easier to move the nucleosomes? I'm asking for a general, not a specific answer, just a general answer. They are attracted to DNA because they are positively charged. What can we do to that charge in order to make them not so strongly attracted? We can neutralize it, right? We can make them less positively charged. Okay, so this is, I think this is just from Wikipedia. It's so this is showing those 4 proteins, 2 H3s, 2 H4s, or actually 6, and the 2 H2s. And this is mapping all the modifications that can take place to these histones. There's many. And this little thing from your, I think that's from your tech, I think it's from this bonus material here. Histone, those are all different ways you can modify the histone proteins in order to modify how they're attracted to DNA. So, this top one is a very important one. Lysines can be acetylated in order to, what's happening to the charge? What's the difference between that and this? This is what charged? Positive. Positively charged. The acetylated lysine is neutral. Okay. So that's going to reduce the affinity for the histone proteins to the DNA whenever it becomes acetylated. This one's a little bit more minor of a modification. This is methylation of arginine. And so you have this arginine, you can add methyl groups. Basically, it adds a little spacer between the positive charge and where it would interact with DNA. So it's going to sort of shield the positive charge from the negatively charged DNA. So it's not eliminating the charge on the arginine, it's just shielding it, or making this more bulky so it can't get as close to the DNA, and therefore it's going to be able to move around. And I'm not going to ask you, there's only one other one up here that I would ask you probably something very specific about. It's straight from 431. Does anyone guess which one of those at the top I might ask you about?
Ubiquitination.
Which one?
Ubiquitination.
That is from 431, but I'm not going to ask you much about it. Does anyone remember what ubiquitin is?
That's the one that doesn't it tag proteins that are misfolded?
So ubiquitin itself is a protein.
Yeah.
That can be added to other proteins. Now, often this is to target them for degradation, but not in this case. In this case, it is just to modify the chromatin by modifying it's not marking these for degradation. And the sumoylation is basically just a slightly different version of ubiquitination. Okay, so which other one of these up there did we talk quite a bit about in 431? Phosphorylation, okay? On what amino acids does phosphorylation take place? I bet you can probably figure that out from this picture. If your eyes are good enough. What kind of amino acids get phosphorylated? What was that?
The ones with hydroxyl groups.
Yes. The ones with alcohols. Okay, which ones?
Serine.
Serine, tyrosine, and threonine. Okay. And I hope you can all draw phosphorylated versions of those amino acids. Okay, so this is just, we're not going to talk about this anymore. This was just like a couple slides. I want to put some of this stuff a little bit in context. So, this is going to be, the way that these histone proteins are going to be modified is going to be different in different types of cells. Right? In different types of cells, right? All of your cells have the same DNA. But things that make different cells different is what genes are going to be turned on and off. And which genes are turned on and off can be basically regulated in two major ways. The first is how is the DNA packaged? Which genes are more accessible to being turned on versus other ones? So in different tissues, your DNA is going to be packaged slightly differently in order to skew gene expression. The second way is going to be transcription factors that bind the DNA to either turn on specific genes or turn off specific genes. Some of those transcription factors can bind the DNA when it's packaged, and other ones really need the DNA to be open and accessible in order to bind it. Now the main topic of the day, chromosome immunoprecipitation, or CHIP. Uppercase C, lowercase h, uppercase I, uppercase p. Okay. This is almost always done when studying eukaryotic cells, although I'm sure you could do something analogous to this if you're studying bacteria. powerful technique for identifying cis elements that specific trans-acting protein is binding to inside the cell. So this is something that's done essentially in vivo. Okay, we're studying the natural binding of a protein to the DNA when it's in the cell. So this is not an in vitro experiment, although after the initial steps of this experiment, then we take it in vitro. But initially, we are assaying the ability of a protein to bind to DNA sequence in its natural environment. I highlighted the word cis elements and transacting protein. Okay? Cis elements refer to sequences of DNA or RNA that are important, usually regulatory. Okay? Transacting factors are proteins that bind to the DNA or RNA. We've kind of already discussed the why questions here. Okay? We already have techniques to look and see whether or not a protein is present in a specific cell or not. But whether or not it's bound to a specific sequence of DNA is what we're going to use this for.
The cis elements, you said that was a sequence of DNA or RNA.
Mm-hmm.
Does that have anything to do with like orientation, like in organic chemistry, cis
No.
and trans?
Cis, in this case, is just DNA or RNA is considered cis. So it's the same. So a protein is something that's from a different region comes into the DNA. So it's pretty much a different terminology.
So like the restriction endonucleases, are those trans because they operate on both sides of the DNA?
Doesn't matter. They're just trans because they're proteins that recognize a cis element. That specific 6-nucleotide element or DNA that they recognize, that's the cis element for those, and then the protein itself is the transacting factor. So it has nothing to do with sidedness in this case.
Oh.
There's no, it's not cis or trans, it's just cis and trans coming in. So yeah, biochemists kind of bastardize those terms for their own purpose. Okay, so here's an overview. This is from the bonus material. You can find different pictures anywhere that's going to describe this. So we start with some cells. You want to be careful about what cells you pick. If you're studying brain, you don't have any brain tissue around, you probably don't want to just pick up some muscle tissue or something else. Okay, you want to make sure you're using the right kind of tissue. And you get the cells, and the first thing we're going to do is we're going to permanently attach the proteins that are bound to the DNA. We want to make that a kind of permanent situation, because we're going to be doing some harsh things to these cells, and we don't want those proteins to come off, especially some of these proteins have very short interactions with the DNA. So we want to capture them. So we're going to take a snapshot in time. And so what we're going to do is we're going to cross-link the DNA and the protein. So any proteins that are bound to the DNA are going to be covalently attached by cross-linking. What reagent have we used in the past for cross-linking?
Glutaraldehyde.
Okay. We're not using that in this case. We're using formaldehyde instead for this. It's a milder cross-linking reagent, and it's easier to reverse, because later we're going to need to reverse it. Okay, and so once we've cross-linked the protein to the DNA, we then lyse the cells, and then we're going to break up the DNA into itty-bitty pieces. You can do this either with sonication, which uses those vibrations at high frequency to break out the DNA into small fragments, or we can use restriction endonucleases. So then we have our fragmented chromatin. You might want to write in here, like write an arrow and say input. Okay, this is our input. Okay. So we're going to have the nucleosomes, but we're also going to have, and this is what they don't really show, I guess they kind of show here. I would draw another protein here, and that's going to be our factor that we're interested in. And then we're going to use antibodies that recognize our protein of interest. And another thing they don't have here is this antibody is probably attached to something. What do you think it's attached to?
Alkaline phosphatase.
Not this time. Immunoprecipitation. What is the antibody attached to an immunoprecipitation?
Like
Or
immobilized.
co-immunoprecipitation? What? Something heavy?
Immobile.
Right? A bead of some sort. Okay? It could be a magnetic bead, or it can be some just regular rose bead or cellulose bead. So then we're going to precipitate it down. We're going to immunoprecipitate. So only the DNA that's attached to our protein of interest should be immunoprecipitated, right? The other DNA should stay suspended in the tube. So we are enriching our sample just with the DNA sequences that are bound to our protein of interest. Yes.
So this is done in a column.
Not as usually just end the centrifuge tube. So we're going to precipitate down by using the bead to pull it down.
Got it.
Okay. Then we're going to get rid of all the protein. You could sometimes use like proteases to help it digest up the protein, but you're also going to use some heat, so you can reverse the formaldehyde cross-linking with heat, and that will liberate our DNA sequences, which we'll then analyze in a variety of ways. We might use some gene-specific primers, or PCR, or quantitative PCR, to try to analyze what these DNA sequences are. Usually you have to have some genes in mind. Usually you're saying, does my protein interact with the gene encoding whatever, right? In that case you can do PCR or quantitative PCR. Or we can take these fragments of DNA and we can clone them sort of randomly into a cloning vector, something like BlueScript. And then we can send those various clones out for sequencing. Or we will talk in a few minutes about using a microarray to analyze which genes were pulled down or what fragments of DNA were pulled down by our approach. Okay, so here it is in word form, kind of the process I just talked about. Okay, first you collect the tissue you want to analyze. That's important that it's the right type of tissue. I mean, if you're fancy, you may care about a very specific cell, like a specific type of cell, and hopefully you have some way, there's cell sorting or FACS, right? And you can actually separate specific types of cells from each other, and then maybe you want to collect those and do this analysis on those. Okay, then we chemically cross-link with formaldehyde, get the DNA and the protein stuck to each other, lyse cells. And then we're going to immunoprecipitate our protein. That's possible. So here's a picture of kind of what's going on. It's possible that, so let's say this is a specific protein, our transcription factor, let's say, some protein that binds DNA and influences transcription. This is our thing. It may not be just bound to the DNA by itself, it may be held in place by other proteins also. So that's another good thing about the cross-linking, is it's going to stabilize this whole thing. So when we immunoprecipitate our protein out, more likely to take place. It's also, because we've cross-linked it, it's possible that this protein's not bound to the DNA at all. It's possible that this protein is associated with the DNA just through protein-protein interactions. Okay, so that's a possible negative thing about this. Later you may have to do something to prove that your protein was specifically bound to that DNA. And next week we'll talk about an experiment that we can actually look at that with. So it could be both direct and indirect interactions with DNA that we're picking up with this technique. Okay, after we've done the immunoprecipitation, pulling it down with the bead, then we use heat to reverse the cross-linking, and then we do the identification of these DNA sequences that we have immunoprecipitated using these variety of techniques that I just discussed. Let's look at this example here, both in the literature. Let's just read the caption for this figure. Okay. Independent chip assay demonstrates binding of, this is the protein. P63 is the name of the protein. I hate these kinds of names for proteins. That just means it's some random protein of 63 kilodaltons. 2 chip DNA sequences. Okay, so chip was performed on this cell line using either an N-terminal RR14 antibody or a C-terminal H129 antibody. So these are antibodies against P63. One of these antibodies recognizes a region on the amino terminus of the protein. The other antibody recognizes the portion of the C-terminal region of P63. Usually you don't see people use two different antibodies. Usually it's just one antibody that recognizes the protein. So they're being extra careful. Okay, enrichment of target genes, which means they picked which genes they wanted to analyze. This is not blind. They are specifically choosing which genes they want to analyze. It's these genes here. Relative to IgG, which we'll discuss in a second on Nagro's gel. Okay, and then they follow this up with qPCR, with the fluorescent qPCR to get absolute values. So this is just for nice looking. This is for actual numerical data. Okay, and so they looked at using. So here's their input. What is input? Remember, I had you write down the word input at one time. So what is this lane acting as?
Fragmented.
What's that?
In fact, fragmented from.
It is, but all of these have DNA that was amplified with PCR. Okay? All three of these lanes are PCR products, whether you see DNA, whether you see a band or not. So the question is, what is their template DNA? That's what's separate in these three lanes for each of these genes. Okay? So all three of these were PCR using primer specific for this gene or for this gene. So the input is a what type of control? So their template DNA is the input. Okay, fragmented chromatin is the input, right? So you take some of the sample out before you continue with this process, and we use this because we have all the DNA here, right? We have all of the DNA. So we do a PCR with some of that to make sure that our PCR works. Which is why when I look at this, I go, I can't believe that they bothered to show some of these. This is really pretty pathetic. There is a band here, but it's really light. So this is a positive control that their PCR conditions are good.
Has to be that there's a band for every single gene.
You should have a band for everything because the genomic DNA is the same in every kind of cell. So if you bust open the cell, collect the DNA, fragment the DNA, and then do PCR on it, you should be able to amplify anything. Right? So this is acting as our positive control for the PCR.
Are they amplifying the entire genome?
No, just small fragments of these genes, of each individual gene.
Oh, because they have digested it prior.
Yes. So this just shows that you didn't lose any of your chromosomal DNA during the early steps. Yes, sir?
Do they remove the protein before they PCR it?
They may have or they may not have. Usually you can do it even if you don't. And they probably digest it with DNase 1 or something like that. That's a good question. I would say don't worry about that small detail. Okay, then we have our second lane. And there's nothing in any of those PCR, right? IgG. So what is that? What is IgG in general? Biology people know what is IgG?
It's an immunoglobulin.
Right? It's an antibody. Okay? This is antibody taken from the same species of animal that whatever antibody you're using. So for your immunoprecipitation, right? Remember you use an antibody specific to your protein. This IgG is just antibodies from an animal that was not immunized against your protein. So it's IgG, none of it recognizes your protein. And what is this showing?
That
Why?
certain things don't bind to your...
That random antibody is not able to pull down, will not immunoprecipitate our protein. Negative controls and positive controls are really important in the kind of work we're talking about.
But that's just one random antibody. There's plenty others that could have
Well, we're taking the whole serum from this animal and using all the antibodies they normally have.
Okay, so it has all the antibodies in that label.
Well, except for that animal was not immunized against your protein of interest. Yes. So a lot of times you take the same. So if you're really good, but most people don't do this anymore. This is old-fashioned the way I used to do it. You would collect serum from your rabbit before you immunize it, and that's what you would use for your IgG here. And then you immunize your mouse or your rabbit, collect your antibody, and then use that for your second one. So it's the same animal that you're using this IgG from. Just one is post-immunization, one's pre-immunization.
Is that a good thing or a bad thing?
It's probably not necessary to worry about it being from the same animal. I think that's why, as we become better and more experienced, we lose some of our superstitions. So it's not important that's from the same animal. Okay, and then finally, they have their lane where they're testing to see whether or not their antibody, and look at all these different things. They're using some very general transcription factor here, so it happens to bind to lots of different genes. Although not this one, and not this one. Now this, I just tell you straight out, this is acting as a negative control. What does that mean? This is what this class is all about. What does that mean? Okay, GAPDH is an enzyme. It is a metabolic enzyme. Do you normally find metabolic enzymes in the nucleus? Is that a very common thing, do you think? Where do you think you find metabolic enzymes?
Cytosol.
In the cytoplasm or in the mitochondria, right? We don't find them very much in the nucleus. Okay. This is not a transcription factor. This is not a protein that binds to DNA. So what are we showing here? If you we're not immunoprecipitating this, right? We're not. Negative control. So, this is something that we're not, our transcription factor is not binding to that gene. In fact, it's possible that gene is even a mitochondrial gene, so I'm not sure.
Is that to show that the protein only binds to genes related to transcription?
No. No, yes, James.
Or does that show that we're not getting random DNA from the cell?
That's right. We're not just getting random DNA. We're getting specific genes to which this P63 binds to. Yeah. All right, good. So I have another simpler example here. Not quite as nice of an example, to be honest. I thought it would be good to see another one. In fact, I was so concerned with this that I put this question. If you were a reviewer for this paper, what questions would you ask, and what would you ask to see? Now, I could also ask the same question for this one. Okay, one of these questions I could also ask for this one. So again, they have a no antibody control. So that they did the no antibody control instead of the IgG control. Which control is better? Right? With an antibody that's just not specific, right? It's closer to resembling the way you're actually doing it. You just don't have an antibody that recognizes a specific protein. So this is a worse control. Now this is still legitimate. This is still done quite often. You still have the beads there, just with no antibody on it. Okay, so their input, which is for PCR control. You see, does that make you worry when your PCR control is negative? That makes me a little concerned. So I wouldn't trust any of the data related to FOXE1. When you compare, does this look weird that these two bands are way up next to the top of this little sliver, a picture they tried to show us? Whereas these are nice and centered in this middle. This is Mary, and looks like there's other bands down here. What would you ask to see if you were the reviewer? The the whole thing, right? Show me the whole gel. But this is, probably 12 different gels that they did this on. This is not all snapshots from the same gel. This is 12 different gels, and they just cut out the part that was relevant. If I was reviewing this, I would say, I would like to see the unaltered data. Please show me your real pictures so I can determine whether I believe these bands. I'd like to see the markers and see that they're the right size bands for what they're doing. I would like to ask them why did they bother to put in the Fox E1 if their positive control lane doesn't even work for PCR. That makes me concerned. Any questions on this? I think I have one more. Oh. Okay, so that's one way of doing the chip assay. And that's probably the way, I would like to maybe do some of those someday, and that's probably how I would end up doing it. However, if you UW, or if you're a research institute like I was at before I came here, and they just got money coming out of everywhere, then you do these kinds of chip assays, chip-on-chip assays. And these chips cost anywhere from $500 a piece to several $1,000s. They used to be like $10,000 a piece. Okay, so it starts out the same. You cross-link the stuff, and you lyse your cells, and you do the immunoprecipitation. All of this is exactly the same. It's only the analysis that's different. Okay? So once we have pulled down our stuff and we release the DNA from the antibody by heating it up or using DNA digestion, then we use an enzyme to put a fluorescent tag on the ends of these DNA sequences. Okay, these are the ones we have immunoprecipitated. Okay. This could be just a fluorescent nucleotide or something that's just enzymatically put on the end of a tag or something. And then you buy this chip, and the chip will have, modern ones have tens of thousands of itty-bitty little spots that you can't see with your naked eye. And they all have known DNA sequences. So this company will make all these DNA sequences, and they'll have a machine that will spot these little DNA sequences on each of these different spots. And then you take your immunoprecipitated DNA and you hybridize it. This is basically like doing a southern blot on a chip. Okay. And so these will anneal to the DNA sequences to which they're complementary. And so you're going to have these fluorescent tags lining up with these specific spots on this chip. And then you just read the chip. Put this in a chip reader. And then it tells you, this gene, this gene, this gene, this gene. It gives you the names of those genes you have immunoprecipitated. This sounds like it's really easy. Okay, it is not. You're going to have many false positives. You're going to have a whole bunch of things glow a little bit. And do you believe them or do you not believe them? And you have to do all the stats, and you have like 10,000 things to do stats on. So this part's the easy part. This part is the hard part, the analyzing the data afterwards. Okay. If you're an expert in it, that's not too bad. But if you're just trying to do this as one part of your project, then it's really tough.
So are those chips made to order with those sequences on them, or is it just full of common?
It's just basically, they don't custom make them for you. So, although they're getting more and more specialized, you could probably buy them from certain animals, or in certain tissues perhaps, or even they're probably going to have like different developmental stages and stuff like that. And they don't have everything on there, right? I mean, especially if you're thinking about most of the genetic material in the cell is not the part that encodes the protein. Most of it's regulatory, or what we call junk still. And so it's pretty daunting to make a gene chip array that will cover every little possibility. So they just do the regions that seem most likely to have proteins bound to them. So there's still bias in it. So this is one form of chip-on-chip. There's a second that's slightly more precise, but it's more effort. I just call this type 2. They're not really called type 1 and type 2. I just did that to separate these two slides. And you do it, and this is from the bonus materials. And it's basically the same thing, except for you also take your input DNA from up there, and then you get rid of the proteins and label it and hybridize it also to the chip. And so that you can compare the intensity of the random DNA versus the stuff we pulled down. So we want to see whether or not specific sequences of DNA are highly enriched compared to this. Okay, so you basically just do compare, and you use different labeling. And so you can hybridize both of these to the chips at the same time. And if this is much more abundant, if your DNA sequence A is much more enriched here, then it should glow with this side 5, which I don't remember if that's red or green. One of the two. So let's say this is yellow, which is probably not. It's probably, let's say it's red. I don't remember. And if your spots turn red, then that means that, oh, it was not immunoprecipitated. But if it turns green, then that means it was much more abundant in this sample than this, and so you can feel confident about it. To be honest, I don't know which of these two methods is more common between the two, because I don't do this kind of stuff.
What would be, I mean, this one has like a positive control with that you're.
Well, with this, hopefully, usually when you're doing this, you have a few genes you already know for sure your protein binds to. And so that would be your positive control. And your negative control could be things like the GAP feature or something like that. You're pretty sure your protein doesn't turn on. Usually with this, you do this to identify candidates, and then you might follow it up with the old-fashioned chip assay with the PCR. Might follow it up with this, or follow it up with the types of experiments we're going to talk about next week, those mobility shift ones. So that's another problem with the chip. You can't just take chip data and publish it. They'll never believe you. You have to back up. You have to pick the ones that were the most. You can publish your whole long list of things you from your chip assay, chip on chip. But then when you want to show important interactions, you're going to have to back that up with additional evidence. All right, a few more minutes. I want to get through one more thing. The variation of chip is written. And this is RNA immunoprecipitation. So it's the same type of thing where we're looking for proteins that bind to RNA. So this is not chromosomes anymore, not chromosomal DNA. It's now RNAs. And here's the basic protocol. I guess not. So you usually want to use an antibody that targets some protein that you either know or that you suspect binds to an RNA. Most frequently, this is mRNAs. And the nice thing about RNAs is they're small enough that we don't have to worry with this whole shearing or cutting up of them. And they're also soluble, so they're going to just take the cell extract and all the RNAs are going to be there. So it says it's unlikely that you have to cross a protein to the RNA. Sometimes that is still done because some protein-RNA interactions are pretty transient, and so you might want to take a snapshot in time. And the other problem is that RNA is not directly sequenceable, right? If we want to sequence RNA or do PCR RNA, we first have to convert it to cDNA. So what do we do to convert mRNA to cDNA? Reverse transcription. Okay. What are the 3 kind of primers that we could use for reverse transcription? Okay, I heard poly-T, which would recognize the poly-A tail if you're doing mRNAs. What's another one? 2, I see you're saying something. Say it out loud. What? I shouldn't slow the degree and stuff. Sorry. Anyone else? What's their second one?
Random something.
Okay. Random hexamers.
There you go.
Okay, was that what you were trying to say? Oh, okay. And gene-specific primers. And then after that, it's basically the same, right, as CHIP. Then you just do new PCR. So you can immunoprecipitate this RNA using an antibody that recognizes some specific protein that you know binds to RNA. And then we're going to do PCR. Or you can probably do rip on chip, I'm sure, too. It's fun to say that. Okay, so here's the question. Assuming that you're doing this, and you have some RNA specific, some primers that are specific to RNA sequence that you're interested in. So let's say you're studying the localization of an RNA inside of a cell. And so it becomes specifically localized, and you have some protein that you think binds to those RNAs that are localized, and you want to demonstrate that in the cell it is able to do that. So here's an example doing that. Okay, so just focus on these first 5 lanes. These are RNAs that become localized in oocytes. If they don't become properly localized, you end up with a 2-headed animal, or hearts on the outside of the body, all kinds of crazy stuff happens if this doesn't happen right. Okay, so here we have first 5 lanes, PCR control. What is that? What term did we already use for our PCR control material? Input, right? This is just RNA isolated from these cells. And then we show that we can do RT-PCR using for this specific RNA. Okay, this experiment was actually pretty complicated. What was done was mRNAs encoding either this protein that binds to these RNAs, this PRRP protein, together with a MIC tag. A MIC tag is sort of like a 6X histidine tag. So we have our PERT protein, and then you just have a small C MIC tag. And this is a common thing that you can buy antibodies against that. Okay? So this mRNA was injected into these cells, and so it made this protein with this PERT and this C MIC. And this PERT bound to the RNA, use antibodies against the C MIC to immunoprecipitate this, and then you do RT-PCR on the RNA to identify it. Okay? So, here's uninjected. Okay? This protein exists normally in these cells. This PERT. So, if we don't inject our PERT with the MIC tag, and we use a MIC antibody, we shouldn't be precipitating this RNA. So that's what this control is doing, is that when we don't inject the MIC tag form of the protein, we don't immunoprecipitate this VG1 RNA. And the control, a negative control that we've seen before, right? The no antibody control. So we do this whole process, and we use the bead without the antibody. Without the antibody, we don't pull down the protein. Without the protein, we don't pull down the RNA. And then we have our positive. Okay? That when we have our MIC-tagged PERT protein and the antibody and everything's present, this protein is able to bind to this RNA, and then we're able to immunoprecipitate it using the MIC tag. And then we have another control. GFP MIC RNA is injected instead. You guys understand the purpose of this? Okay. Instead of having our protein that we know, that we think binds this RNA, we replace it with some protein that definitely does not bind to RNA at all. GFP is green fluorescent protein. It does not bind to RNA. And so if we replace the PERT with GFP, it doesn't bind the RNA, and then we do this whole immunoprecipitation process. This demonstrates that the whole process, the injecting of the RNA, the presence of a MIC tag on a protein, the presence of these beads and the antibody, when all of these things are there, we still do not immunoprecipitate this VG1 RNA. So it's sort of the ultimate control. Everything's the same except for the protein. Okay, and all these are just looking at different RNAs. So I just answered all these questions. I think that's it. Do you have any other questions?